You are watching single-cell multiome analysis series with SciDAP. It is our 12th tutorial video and we are continuing the analysis of the example dataset using the single-cell multiome pipeline. We already finished dimensionality reduction step for RNA part and now we will start the clustering. Let's add a new analysis and select the single-cell RNA cluster analysis pipeline. We could call it step 4 and give it a new name. Now we need to select from the previous step, target dimensionality, and the clustering resolution. Clustering resolution will modify the number of clusters you will get at the end. Sometimes it is hard to assign the cell types to clusters if the resolution is too high, so you may need to use multiple resolutions to see which one fits your data better. We can provide here multiple values. Let's try 0.3, 0.4, and 0.5. Keep in mind that if you put too many, it may affect the speed of the processing. Fine-tuning markers is a very useful option for manual cell type annotation of the clusters. You could type any genes that you think are helpful for this purpose. I will put just a few. Let's also put CDLA4. You could upload a GMT file. File could be obtained from GZA website. If you would like to run multiple at the same time, you would need to format them. First, you open the GMT file as a text file, and then just copy paste this in Excel. Here I already saved an example file with 4 gene sets combined. This is 4 GMT files together that I copy pasted to save some time. And this is how it's formatted. It is the name, the source, and the gene sets. All this information is available when you open the GMT file as a plain text. Then you could save it as a TSV and then provide it as an input for your analysis. In the next video, we will review the results of this clustering.